Have you ever stopped to think about that invisible conversation, the one happening inside your body, like
Mhm.
right now? Billions of cells constantly talking.
It really is. It's basically the language of life itself, isn't it? This constant back and forth, keeping everything running smoothly, maintaining that internal balance, homeostasis.
Without it, everything just grinds to a halt.
Exactly.
And that's exactly our plan for this deep dive. We're getting into cell signaling in physiology, straight from Chapter 5 of Vander's Human Physiology.
Yeah. The core
Right.
mechanisms.
Our goal is to really sort out the difference between intercellular,
Cells talking to each other.
yeah, and intracellular, the messages inside a single cell. We'll look at the chemical messengers, the pathways, the whole setup.
And understanding these basics really helps you grasp how your nervous system, your endocrine system, well, how everything works. It all hinges on this communication.
So we'll start with the cell's ears, the receptors, then look at how signals get processed, the pathways.
And importantly, how the signals stop, how the conversation ends.
It's going to be fascinating. Let's jump in.
Okay.
All right, first things first. How does a cell even, hear a message? We need to talk about receptors.
Right. Receptors are the detection system. Think of the chemical messengers, we call them ligands, as the actual signals being sent out.
And the receptors are specific proteins waiting on the target cells designed to grab onto those signals.
Exactly. And that grabbing, that binding, it's not passive. It actually changes the receptor's shape. It's conformation.
And that shape change is the activation.
That's the activation. It flips the switch, basically, and that kicks off a whole cascade of events inside the cell, leading to the actual response, a whole process. That's signal transduction.
The signal is the activation, transduction is turning it into action.
You got it. Signal in, response out via that transduction pathway.
Okay, so where are these receptors? Are they all in one place?
Mostly two main locations. The most common type are plasma membrane receptors. They sit right on the outer surface of the cell, like little antennas.
Sticking out into the environment around the cell.
Precisely. They're transmembrane proteins, meaning they go all the way through the membrane. Part sticks out to catch messengers, part sticks inside to start the signal relay.
And these are mainly for messengers that can't get through the membrane easily.
Exactly. For water-soluble messengers, the cell membrane is fatty, a lipid bilayer, so water-soluble stuff just can't diffuse across. These receptors provide the way in without the messenger ever having to cross.
The book uses a great analogy, like a lock on a fortress wall. The messenger is the key.
It fits the lock on the outside, turns it.
And that triggers mechanisms inside the fortress. The key itself doesn't need to go through the wall.
Perfect picture for those water-soluble signals, but then you have messengers that can slip through the membrane.
The lipid-soluble ones.
Right.
Like steroid hormones, thyroid hormone.
Right. For those, the cell has intracellular receptors. They're inside the cell, floating in the cytosol maybe, or sometimes right in the nucleus.
And the big difference here is how they work.
Yeah.
Right. They interact directly with DNA.
That's a key distinction. Unlike the membrane receptors, these intracellular ones often have a part that binds directly to DNA. So they transduce signals by directly messing with gene expression, turning genes on or off.
Wow. Okay, so direct genetic control versus relaying a signal from the outside. Two very different strategies.
Definitely. Based entirely on whether the messenger can get inside the cell on its own.
Okay, so we know what receptors are and where they are. Now let's dig into how that interaction between the ligand, the messenger, and the receptor actually works. There are four key features mentioned: specificity, affinity, saturation, and competition.
Yeah, these four really define the interaction. Let's start with specificity.
This sounds like the lock and key idea again.
It is, essentially. A receptor is usually very picky. It binds only one specific type of messenger, or maybe a small group of messengers that look very similar structurally.
So a cell only responds to a signal if it has the right receptor for that signal.
Exactly. It's like a cellular zip code. That's why I say norepinephrine can affect heart cells and brain cells, because both types of cells have the right kind of norepinephrine receptor, but the response might be different in each cell type.
Ah, so the receptor determines if the cell listens, but the cell type determines what it does in response.
Precisely. Specificity ensures the message gets to the right address, but the address itself determines the action taken.
Okay, next up. Affinity. What does that refer to?
Affinity is just the strength of the binding. How tightly does the messenger stick to the receptor?
Like a strong magnet versus a weak one.
Good analogy. High affinity means strong binding. Even if there aren't many messenger molecules around, a high affinity receptor can grab them effectively.
Which would be important for drugs, I imagine. Needing less drug if the affinity is high.
Absolutely. It means a lower dose might be just as effective, which often means fewer side effects. It's a critical factor in pharmacology.
Makes sense. Then there's saturation. What limits are we talking about here?
Well, a cell only has so many receptors for any given messenger, a finite number.
Like parking spots in a lot.
Exactly like that. As you increase the messenger concentration, more and more receptors get filled, more parking spots get taken, and the cell's response gets stronger.
But eventually.
Eventually, all the receptors are occupied, the parking lot is full, you've reached saturation. At that point, adding more messenger doesn't increase the response any further. You've hit the maximum.
The cell is doing all it can based on that signal pathway.
Right. The system is maxed out.
Okay, last one. Competition. This sounds like molecules fighting over a receptor.
That's exactly what it is. If you have different molecules that are similar enough in shape, they might all try to bind to the same receptor site.
It's also huge for medicine, right?
Foundational. This
Yeah.
is where antagonists and agonists come in.
Okay, break those down. Antagonists first.
Antagonists are competitors. They bind to the receptor, taking up the spot, but they don't activate it. They just block the natural messenger from binding and doing its job.
Like putting the wrong key in the lock, it fits, but it won't turn, and it stops the right key from getting in.
Perfect. Beta blockers are a classic example. They block epinephrine and norepinephrine receptors on the heart, lowering blood pressure. Antihistamines block histamine receptors to stop allergy symptoms.
Okay, so antagonists block. What about agonists?
Agonists also compete and bind to the receptor, but they do activate it. They mimic the action of the natural messenger.
So they're like a mimic key that actually works.
Pretty much. Decongestants like phenylephrine are good examples. They mimic epinephrine at certain receptors to constrict blood vessels in your nose, which helps clear things up. They activate the receptor just like the natural signal would.
Specificity, affinity, saturation, competition. Got it. 4 rules of engagement for messengers and receptors.
They dictate who talks to whom, how strongly, for how long, and who else might interfere.
Now, these receptors, they aren't just fixed things, are they? The cell can actually change how many receptors it has.
Absolutely. This is regulation of receptors, and it's incredibly important for keeping things balanced homeostasis again. The number of receptors and even their affinity can change physiologically.
Okay, so how does that work? Let's talk about downregulation.
Downregulation happens when a cell is bombarded with a high concentration of a messenger for a prolonged time. The cell basically says, okay, this is too loud, and it reduces the number of receptors for that specific messenger.
It turns down the volume on the signal.
Exactly. It desensitizes itself. This often involves pulling the receptors inside the cell, a process called receptor-mediated endocytosis, and then breaking them down. It's a local negative feedback loop.
Makes sense. Protects the cell from overstimulation. What about the opposite? Upregulation.
That happens when the messenger concentration is chronically low. The cell wants to be more sensitive, so it increases the number of receptors for that messenger.
Turning the volume up to catch a faint signal.
Precisely. It might involve moving pre-made receptors stored inside the cell in vesicles out to the plasma membrane.
Is there a common example of this?
A classic one is denervation supersensitivity. If the nerve supply to a muscle is damaged, the muscle cells get less neurotransmitter signal. So they upregulate their receptors, becoming hypersensitive to any small amount of neurotransmitter that is still around.
So both down and upregulation are ways the cell adapts to changing signal levels, trying to keep its response in a normal range.
Exactly. Perfect examples of homeostasis at the cellular level, adapting to maintain stability.
Okay, we've covered how cells detect signals via receptors and how those interactions are governed and regulated. Now let's get into the nitty-gritty. What happens after the receptor is activated? The signal transduction pathways.
Right. Receptor activation is just the starting gun. The signal transduction pathway is the whole race that follows, linking that initial binding event to the cell's final response.
And the responses can be incredibly varied, can't they?
Hugely varied. Changes in membrane permeability, electrical state, metabolism shifts, secretion changes, starting or stopping cell division, muscle contraction, lots of possibilities.
But you said earlier it all boils down to changing proteins.
Ultimately, yes. All those diverse responses come from altering specific cell proteins, changing their shape, activity, location, or how many of them are made.
And the pathway depends heavily on whether the messenger was lipid-soluble or water-soluble because of where their receptors are.
Correct. Let's tackle the lipid-soluble messengers first. Remember, these guys can walk right into the cell.
Steroid hormones, thyroid hormone, their receptors are usually inside the intracellular or nuclear receptors.
Exactly. So the pathway is quite direct. The messenger diffuses in, finds its receptor inside the cytosol or nucleus, and binds.
Activating the receptor.
Right. And this activated messenger receptor complex then acts as a transcription factor.
Meaning it directly controls genes.
Yes. It binds to specific regulatory regions on the DNA and typically either increases or decreases the transcription of specific genes into messenger RNA.
And that mRNA then goes off to the ribosomes.
And directs the synthesis of new proteins. So the end result is a change in the concentration of specific proteins within the cell, and that change is what causes the cell's response.
Like cortisol turning on genes for metabolism or turning off genes for inflammation.
Precisely. It's a relatively slower process because it involves making new proteins, but it can have very profound and long-lasting effects.
Okay, that seems pretty direct. What about the water-soluble messengers, the ones stuck outside the cell membrane?
Now things get a bit more complex involving relays. Since they can't get in, they bind to plasma membrane receptors. We need some key terms here.
Okay.
The initial messenger outside the cell is the first messenger.
Hormone, neurotransmitter, whatever it is.
Right. Because it can't get in, its message needs to be relayed inside. The molecules generated inside the cell in response are called second messengers.
They carry the signal from the membrane deeper into the cell.
Exactly. And very often, these pathways involve enzymes called protein kinases.
We mentioned these. They add phosphate groups to proteins.
Yes, using ATP as the source.
Hmm.
Phosphorylation by a kinase often acts like an on or off switch for the target protein. Kinases often work in cascades. One activates the next, which activates the next, amplifying the signal.
And there must be a way to turn them off, too.
Absolutely. Protein phosphatase are the enzymes that remove those phosphate groups, reversing the kinase's action and helping to shut down the signal.
Okay, first messenger, second messenger, kinases, phosphatases.
Yeah.
Got players. Now, what about the different types of membrane receptors for these water-soluble guys? There were 4 main classes.
4 main classes. First, receptors that are ligand-gated ion channels.
So the receptor is the channel.
Correct. The protein structure acts as both the receptor and the pore through the membrane. When the first messenger binds, the channel physically opens or closes.
Letting specific ions flood in or out, like calcium.
Exactly. It causes a very rapid change in the ion flux across the membrane, which can change the cell's membrane potential or trigger other calcium-dependent events almost instantly.
Super fast response, like flipping a switch for ions.
Very direct, very fast. Second class, receptors that function as enzymes. The most common type here are the receptor tyrosine kinases.
So the receptor itself has enzyme activity built in.
Yes, on the part that sticks inside the cell. When the messenger binds outside, the intracellular part gets switched on specifically, its tyrosine kinase activity.
What does it do then?
It typically phosphorylates itself autophosphorylation on specific tyrosine residues.
It adds phosphate tags to itself.
Right. And those phosphotyrosines then act like docking sites, recruiting other cytoplasmic proteins, which then get activated, often by being phosphorylated themselves, and carry the signal onward.
So receptor activation leads to self-tagging, which leads to docking and activation of downstream players.
That's the essence of it. There are variations, like receptors that act as guanylyl cyclists, making cyclic GMP, cGMP as a second messenger, and nitric oxide, NO, a gas, can actually diffuse into the cell and activate a cytosolic granulocyclase, also making cGMP, important in blood vessel relaxation.
Okay, third class.
Receptors that interact with cytoplasmic Janus kinases, or JAKs. Here, the receptor doesn't have its own enzyme activity.
So it needs a partner enzyme.
Exactly. It's closely associated with a separate tyrosine kinase enzyme from the JAK family, located just inside the membrane. Messenger binding activates the receptor, which then activates its associated JK.
And the JK then does the phosphorylating.
Yes. The activated JK phosphorylates various target proteins, including many transcription factors, which then travel to the nucleus to alter gene expression, important for signals like cytokines in the immune system.
So similar outcome to the lipid-soluble pathway changing gene expression, but via a different route starting at the membrane.
Right, an external signal leading to internal gene changes, mediated by JKs.
And the fourth, and you said biggest class.
The G-protein-coupled receptors, or GPCRs, huge family, involved in countless processes.
What's the G protein part?
The receptor isn't directly linked to an enzyme or channel. Instead, it's coupled to an intermediary protein complex located on the inner membrane surface called a G protein.
An intermediary, like a middleman.
Exactly. G proteins have 3 subunits: alpha, beta, and gamma. The alpha subunit is the key player here. It can bind GDP,
Mm.
inactive state, or GTP, active state.
So how does it work? Messenger binds the GPCR.
The GPCR changes shape and activates the G protein. Specifically, it causes the alpha subunit to release its GDP and bind GTP.
That's the switch flicking on.
Right. The GTP-bound alpha subunit then detaches from the beta and gamma subunits and moves along the membrane until it bumps into an effector protein.
And the effector protein is?
It could be an ion channel or an enzyme. The activated alpha subunit interacts with this effector, either opening, closing the channel, or activating, inhibiting the enzyme.
So the G protein links the receptor to the next step, the effector.
It's the crucial link. And that effector then generates the next part of the signal, maybe changing ion flow or producing second messengers.
How does it switch on?
The alpha subunit has its own internal timer. It naturally hydrolyzes GTP back to GDP. It has GT-pace activity. Once it's back to GDP, it lets go of the effector and rejoins its beta and gamma partners, resetting the system.
Wow, okay. 4 distinct ways water-soluble messengers can get their signal across the membrane. Channels, enzyme receptors, JAK-linked receptors, and these GPCRs.
Each activating different downstream pathways.
Let's focus on those downstream pathways now, especially the second messengers generated by some of these systems, like the GPCRs. Cyclic AMP, GMP seems like a major way.
Absolutely. Classic second messenger. It's generated when a first messenger activates a GPCR coupled to a stimulatory G protein called G.
G is for stimulatory. Makes sense.
This activated GL protein then turns on the effector enzyme adenyl cyclase, which is embedded in the membrane.
And adenyl cyclase makes CAMP from what?
From ATP. It cyclizes ATP into cyclic AMP. Lots of it, potentially.
Okay, so now you have a bunch of CAMP inside the cell. What does it do?
CAMP diffuses through the cytosol, and its main job is to activate CAMP-dependent protein kinase, also known as protein kinase A.
PKA, another kinase. So
Yeah.
PKA then phosphorylates things.
Exactly. PKA phosphorylates a whole range of target proteins, enzymes, channels, even transcription factors, and that phosphorylation dictates the cell's ultimate response to the original first messenger.
How's the CAMP signal turned off?
An enzyme called CAMP phosphodiesterase is constantly active in the cytosol, breaking down CAMP into inactive AMP. So as soon as adenyl cyclase stops making it, the phosphodiesterase quickly clears it out.
You mentioned caffeine inhibits this phosphodiesterase.
That's right. Caffeine and theophylline keep CAMP levels higher for longer by slowing its breakdown. That contributes to their stimulant effects.
And this CAMP pathway is famous for signal amplification, right?
Phenomenal amplification. One messenger molecule binds one receptor, activates maybe a few G proteins. Each G protein activates one adenyl cyclase. But that one enzyme can churn out hundreds or thousands of CAMP molecules.
And each CAMP activates a PKA.
And each PKA can phosphorylate hundreds or thousands of target proteins. The signal gets massively amplified at each step. You mentioned the epinephrine example earlier, one molecule leading to 100 million glucose molecules released. That's amplification.
Incredible. Allows for huge responses from tiny initial signals. And PKA can phosphorylate different targets in different cells.
Yes, which explains how the same hormone, like epinephrine, can cause fat breakdown in adipose cells, but glucose release in liver cells. Different targets for PKA. PKA can even go into the nucleus and phosphorylate transcription factors.
But G proteins aren't always stimulatory, you said. There's G.
Right. Some GPCRs couple to an inhibitory G protein, G. When activated, G inhibits adenyl cyclase, lowering CAMP levels and damping down the cell's response. It provides a way to put the brakes on.
So you have accelerators, G, and brakes to controlling CP levels. Nice balance.
Exactly. Fine-tuning the response.
Okay, what's another major second messenger system involving GPCRs? The book talks about phospholipase C, DAG, and IP3.
Yes, another crucial pathway. This one is typically activated by GPCRs coupled to a different type of G protein called GQ.
GQ, okay. What does GQ activate?
It activates the plasma membrane effector enzyme phospholipase C, PLC.
And PLC does what?
PLC targets a specific membrane phospholipid called PIT2, phosphatidylanicetyl 4-carboxybiphosphate. It cleaves PIP2 into two separate molecules, both of which act as second messengers, diacylglycerol, DAG, and inosyltriphosphate, IP3.
2 second messengers for one reaction.
Yes. DAG is lipid-soluble, so it stays in the membrane. Its main job is to recruit and activate another kinase called protein kinase C, or PKC. PKC then phosphorylates its own set of target proteins.
Okay, so DAG activates PKC in the membrane. What about IP3?
IP3 is water-soluble, so it diffuses away from the membrane into the cytosol. It travels to the endoplasmic reticulum, ER, which is a major storage site for calcium ions within the cell.
And what happens at the ER?
IP3 binds to specific IP3 receptors on the ER membrane. These receptors are ligand-gated She2+ channels.
So IP3 binding opens calcium channels on the ER.
Precisely. This causes a rapid release of stored He2+ from the ER into the cytosol, leading to a sharp spike in the intercellular calcium concentration.
Which brings us neatly to K2 plus itself as a second messenger.
Indeed, calcium is hugely important. Cytosolic levels are normally kept incredibly low by pumps that push it out of the cell or into the ER, but signals can cause it to rise dramatically.
Through IP3 opening ER channels, as we just saw. Any other ways?
Sure, some plasma membrane channels can let C2+ in from outside, maybe ligand-gated channels or voltage-gated channels in excitable cells. Or inhibiting the pumps that remove C2+ can also raise levels.
Okay, so cytosolic calcium goes up. How does it act as a signal?
Calcium exerts its effects by binding to various calcium-binding proteins in the cytosol. Binding changes their shape and activates them.
The most famous one is probably calmodulin.
Absolutely. Calmodulin is ubiquitous. When calcium levels rise, calcium binds to calmodulin, activating it.
And what does the activated calmodulin complex do?
It then interacts with and activates or inhibits a whole host of other proteins, including a very important class called calmodulin-dependent protein kinases.
More kinases. Phosphorylation again.
Often, yes. These kinases phosphorylate target proteins to produce the cell's response. But calcium can also act more directly, like binding to troponin in muscle to trigger contraction, so it's incredibly versatile.
So CMP and the CANA2 plus DGAP3 system are two major second messenger pathways, often triggered by GPCRs, leading to kinase activation and phosphorylation cascades.
That's a great summary of two really central pathways.
Let's touch briefly on another group mentioned, the eicosanoids. These sound different.
They are a bit different. They're not proteins or amino acids. They're lipid derivatives. Specifically, they're derived from arachidonic acid, a fatty acid that's part of the cell membrane phospholipids.
Includes things like prostaglandins, thromboxanes, leukotrienes.
That's right. The whole process starts when an enzyme called phospholipase A2 gets activated, usually by some stimulus, and snips arachidonic acid out of the membrane.
Then what happens to the arachidonic acid?
It can go down two main enzymatic pathways. The cyclooxygenase, COX pathway, produces cyclic endoperoxides, which are then converted into prostaglandins and thromboxanes.
Okay, and the other pathway?
The lipoxygenase pathway, which converts arachidonic acid into leukotrienes.
What do these eicosanoids generally do?
They tend to act locally as paracrine signals affecting nearby cells or autocrine signals affecting the cell that made them. Sometimes even intracellularly. They're involved in a huge range of things, but especially blood vessel function and inflammation. They're usually broken down quickly.
And the drug connection here is with things like aspirin.
Exactly. Aspirin and other NSAIDs, nonsteroidal anti-inflammatory drugs, work by inhibiting the COX enzyme. By blocking COX, they block the production of prostaglandins and thromboxanes, which helps reduce pain, fever, and inflammation associated with those molecules.
What about steroid anti-inflammatories?
They work further upstream. They typically cause the production of a protein that inhibits phospholipase A2, so they block the release of arachidonic acid itself, thereby shutting down the production of all eicosanoids from both the COX and lipoxygenase pathways. That's why they're generally more potent anti-inflammatories.
Okay, fascinating connection to everyday drugs.
Yeah.
Finally, we need to talk about turning signals off. Cessation of activity. These pathways can't just stay on forever.
Absolutely not. Responses are typically transient. The signal needs to stop for the cell to be able to respond to new signals and maintain control.
How does the signal get terminated? What are the main mechanisms?
Several things have to happen. First, the first messenger concentration usually drops. The messenger might get metabolized by enzymes nearby, taken up by cells and destroyed, or simply diffuse away. Less messenger means less receptor binding.
The initial trigger fades away.
Right. And as the first messenger decreases, second messenger production slows down, and existing second messengers like CAMP or CA2+ are actively removed or broken down by enzymes like phosphodiesterase or calcium pumps.
But can the receptor itself be shut down, even if the messenger is still around?
Yes, that's crucial too. Receptor inactivation can happen in a few ways. The receptor might be chemically altered, often by phosphorylation, which can lower its affinity for the messenger, causing it to pop off.
Like making the lock slightly sticky so the key falls out easier?
Kind of. Or phosphorylation might prevent the receptor from coupling to its G protein, breaking that link.
Uncoupling the system.
Exactly. And another major way is internalization. The cell can literally pull the bound receptor messenger complex inside via endocytosis, taking it off the surface entirely.
Removing the detector from the surface.
Right. All these mechanisms ensure the signal is temporary and controllable. Often, the signaling pathway itself triggers negative feedback loops that promote its own inactivation.
It's self-limiting.
It has to be. And remember, cells aren't just dealing with one signal at a time. They're integrating inputs from many different messengers simultaneously. There's loads of crosstalk between these pathways, making the actual response a highly integrated outcome of all the signals being received. It's incredibly complex.
A real symphony, like we said at the
For
start.
sure.
Okay, what a journey through cell signaling. From receptors acting as locks and keys to the rules of affinity and saturation, the dynamic regulation of receptor numbers.
And diving into the pathways, the direct action of lipid-soluble messengers on genes, and the complex relays involving first and second messengers, G proteins, kinases, CAMP, calcium, DG, IP3 for the water-soluble ones.
Not forgetting eicosanoids and how signals are precisely turned off. We've really unpacked the fundamental language cells use to communicate.
It truly underlies almost everything your body does.
So let's leave our listeners with a final thought. We've seen how intricate and, frankly, how robust these systems usually are, but also how delicate they can be. You mentioned pseudohypoparathyroidism earlier, where a defect in one component, the GS-alpha subunit, causes widespread issues, calcium problems, thyroid issues, bone development problems.
It highlights the interconnectedness. A single faulty link in one common pathway can have ripples across multiple organ systems. So thinking about that, as we learn more and more about these pathways, these intricate details, how might this knowledge change things? How could we potentially leverage this understanding, not just to fix broken signals and diseases, but maybe even to design new ways for cells to communicate? Could we enhance healing or recovery by sort of orchestrating cellular conversations in novel ways? What are the possibilities when we truly master this language of life?